Hello everyone, I am Dr. Charu Dogra Rawat, Professor at Ramjas College, University of Delhi, and I have been running a session on biotechnology. The topic for today's would be production of cloned and transgenic animals that we continue with our previous session. The specific learning objective for today's session would be to understand the method of production of transgenic animals. In predominantly, we will talk about the pronuclear microinjection. The references used for the educational purposes is primarily the T.A. Brown, Gene Cloning and DNA Analysis, and Glick and Pasternak patent, Molecular Biotechnology Principles and Applications of Recombinant DNA. Figures, etc. would be taken from these books until and unless stated in the slides. Now, we have been discussing that the objective of genetic engineering is often to create a useful product or a commercial process. And one of its uses and applications is the production of cloned or transgenic animals. We have talked about the basic recombinant DNA technology, the techniques involved in that, as well as the advanced techniques such as the gene editing tools, etc. So, the major application of this genetic engineering or the recombinant DNA technology is the production of cloned or transgenic animals. Now, what is a transgenic animal? A transgenic animal is one that has integrated a gene or a DNA sequence, which we call a transgene, that has been transferred by human intervention into the genome of a cell. Now, this transgene is the gene of interest, our interest that we want to study for its expression, or how, what are basically the structure as well as the function of these transgenes. So, that is the gene of interest that we introduce into a host organism, into the genome of a cell, primarily by human intervention. In other words, a transgenic animal can also be defined as that which has stably incorporated the transgene into its germline. Now, this is very important because a transgenic line should have this transgene expressed in its all the generations. So, it is able to pass the transgene onto its offspring. So, that is a transgenic animal that we want to develop. Now, very simple strategy that we had already discussed of generating these transgenic animals is that first you clone a gene. So, that cloning a gene involves the recombinant DNA technology, where we have talked about how the genes of interest can be cloned into vectors and introduced into various host cells, the bacterial cells, yeast cells, or or through various mechanisms, even in eukaryotic cell lines, and then how they can be stably maintained in these cell lines. They can be screened first of all, that which cells have a gene of interest, and then they can be stably maintained into it. So, very simple strategy to start with for creating a transgenic animal is to identify and isolate our gene of interest and then clone it into a suitable vector and introduce it into a suitable host. Now, this for cloning, for molecular cloning, and then we again isolate this vector or the cloned gene, and then it is injected into the nucleus of a fertilized egg. Now, these inoculated fertilized eggs are then implanted into a receptive female or the foster mothers for successful completion of the mammalian embryonic development, because we cannot, the fertilized eggs cannot be developed outside the mammalian body. So, it has to undergo that mammalian embryonic development, for which we introduce these fertilized eggs containing our transgene into the foster mothers. The third step is basically that some of the offsprings derived from the implanted eggs carry the cloned gene in all of their cells, and these are called as the founder organisms. And then these founder organisms are bred, first of all they are identified and then they are bred to establish the new genetic lines, and these are then the transgenic lines which is expressing or containing our cloned gene integrated into their germline. So, this is a very simple strategy for any of the methods that is used for creating a transgenic animal. Now, these are the different mechanisms that we have talked about. The first one is the viral mediated gene transfer, which we have talked about in detail in our previous session, where we talked about the retroviral vectors, how the genes are cloned into the retroviral vectors and then introduced into the host cells, into the bacterial cells, or into the mice cells for creating the transgenic animals. Today we will talk about the DNA microinjection, and primarily in that we will talk about the pronuclear microinjection. In our subsequent sessions, we will talk about the embryonic stem cell usage for creation of the transgenic animals, as well as we will talk about the other different methods such as the nuclear transplantation or the introduction of transgenes into the sperms or the gametes or even the gonads. So, we will talk about all these methods in a subsequent session. Today, we will talk about the pronuclear microinjection. Now, pronuclear microinjection, the steps involved are basically that first of all, the number of available fertilized eggs that are to be inoculated is increased by stimulating donor females to superovulate. So, first step is superovulation. You know, a normal female will release or will form and release these eggs in a certain amount of number. You know, there's a certain number that every species, particularly we'll talk about mice, but then the cloning is also done in sheep or cattle, cow. It is also done in there, and at the end we'll just do a comparative account of what are the reproductive requirements or strategies of these different animals. But in general, whatever the number of eggs they are producing, we have to superovulate them so that we can get a large number of, or at least more number of fertilized eggs that they normally produced. So, first basically step is to stimulate the donor females to superovulate. Now, female mice are given an initial injection of pregnant mare serum and another injection about 48 hours later of human chorionic gonadotropin. Now, how we are, as I said, we will be talking about the methodology in mice. We will be taking another example also, that how the transgenics are created. So, we take the female mice, the donor mice, a healthy donor mice, and we give it the different injections. So, these are the different injections as you can see. So, there is a female that we want to, that is the donor female, and in that donor female we first inject it with what is called as the pregnant mare's serum. So, these PMSG we are going to inject it, and then after 48 hours we inject the human chorionic gonadotropin, after which the mating is done. Now, mating is done with a male, which is basically, this is a normal female-male mating. The only thing is that the female has been superovulating to generate a large number of eggs. Simultaneously, we also, breed two other pair, which contains a female, which is actually we make it a pseudo pregnant female, because it has to have the same conditions as this female, because we will take the egg from the donor, and this is the foster mother. So, the foster mother also, we basically follow the same path. We do not inject it because we don't want it to superovulate, but we simultaneously run this breeding so that the pseudo pregnant females are basically of the same time as we are getting the donor female mating done. And we use the vasectomized male mice. So, vasectomized male mice do not produce any sperms. So, it is going to make the female pregnant. That is why it is called as a pseudo pregnancy, because the mating will occur, but because it does not produce any sperms, so there will not be any pregnancy that occurs, but it is undergoing the same cycle and same mechanism as the donor mother. So, this is the superovulation, and after superovulation, as you can see in these figures, these photos, they select the mice by the occurrence of this vaginal plug. So, the fertilized females will have this plug, and the non-fertilized females do not have this plug. So, they can identify the females that have mated. So, in both the pairs which has bred, the superovulated female, which we have created by the injection of these hormones, the mare serum as well as the HCG, human chorionic gonadotropin, so that it is superovulating. So, when we mate them, then we identify the mated females, that there is a vaginal plug in these females as apart from them. So, we separate them. So, this is basically how the superovulation is done. These photographs show the mechanism of vasectomy. So as I said that we need to get a male which is vasectomized so that it is not producing any sperm. So this is how basically it is done. So first of all, from the abdominal side, pressing is done so that the testes come into the scrotal sacs. Then in the scrotal sacs, an incision is made, and then vas deferens is pulled out, and then vas deferens is kept in place, and as you can see in this case, so it makes a V-shaped structure, and then from there these vas deferens are cut from right side as well as the left vas deferens. So both the vas deferens are then cut so that there is no sperm production. So it is very essential and a part of the creation of transgenic mice, because we want to get a foster mother which has mated at the same time as the donor female, but we don't want it to be pregnant. So it need to go the mating process, and therefore it will not get pregnant because the male is vasectomized and is not able to produce the sperm. So these are the two mechanisms by which the superovulation is done and the vasectomy is done. These superovulated mice produces about 35 eggs instead of the normal 5 to 10. So as I said, superovulation means that the more number of eggs are produced. So these females which have been given hormonal treatments as stated earlier would produce around 30-35 eggs as compared to 5 to 10 eggs. Superovulated females are then mated so that eggs become fertilized. Now, why do we need superovulation? Because we will see that the overall efficiency of production of transgenic animals is not very high. And at each step, we'll subsequently see that it is around only 5% of the eggs that we take, they become, they are able to produce the transgenic mice. So the overall efficiency is very less. So we cannot start with the starting material. We should have a large number of eggs, because at every step of creating a transgenic mice, the efficiency will fall. So when we have large number of eggs to start with, there will be a more probability of creating the fertilized egg, and then there will be a more probability, because as I said, at every step the efficiency is going to be reduced. So we superovulate the female so that to start with we have a large number of eggs that can be fertilized, and then subsequently at the end at least we are able to produce the transgenic mice. So these superovulated females are then mated so that the eggs become fertilized, and then they are killed. So we will just take these fertilized eggs and will kill the female mice. The fertilized eggs are then flushed from their oviducts. And how do we do that? This is how the harvesting of the fertilized eggs take place. So you can see that first of all it is deskinned, the females are deskinned, and then the incision is made, and the oviduct along with the ovary, it is pulled out. Then the first cut is made between this the oviduct and the ovary. So this cut is made, and then again the uterus is also pulled out, and then the second cut is made at the uterus, and then from this uterus the fertilized eggs are basically flushed out. So this is how basically it happens. So the first of all the ovary and the oviduct is cut, and then at the uterus it is pulled out and again cut, so that the from the oviduct to the uterus, the entire eggs can be flushed out. So this is how the harvesting of the fertilized egg then takes place. Now these show some micrographs, the freeze fracture micrographs, where you can see that these eggs have been fertilized eggs, or the zygotes have been harvested, and they contain cumulus along with it. So these are the cumulus region, and these are the fertilized eggs. They are then treated with hyaluronidase, which basically degrades all this cumulus, and these are the isolated the separated zygotes, as you can see. Now the arrows represent basically the mature zygotes, these are the full zygotes, but here you can see just with the arrowheads that these zygotes have been degenerated. So as I said, every step there is a loss of efficiency. So you get a large number of eggs, so you superovulate the females, and then when you isolate the eggs, then also there is a when you are then there is a loss of efficiency during fertilization, because whatever the number of ovulated eggs are, all will not be fertilized, and then when you isolate them, there will be loss in number in the isolation part, and as you can see here, then there will be a degeneration of the zygotes as well. So zygotes are harvested with follicular cumulus cells to start with, and then after successive treatments with hyaluronidase, the normal zygote will be isolated. And as you can see, these are the ones and these are the ones which are degenerated. So that is how these zygotes are then harvested. The next steps involves the micro injection of the fertilized eggs. Now this is the micro injection part. So we have got now a fertilized egg, and in this fertilized egg we need to introduce our gene of interest, that is the transgene. Now these fertilized eggs usually occurs immediately after their collection. The micro injected transgene construct is often in a linear form and free of prokaryotic vector DNA sequences. So this is again very important, When we studied recombinant DNA technology, we studied that to increase the number of the, to increase the number of the clones, the number of the gene of interest, we do molecular cloning, in which we clone this gene of interest into the vector DNA and then introduce the vector DNA into a suitable host, generally bacterial host, that multiplies, and therefore the transgene also, or the gene of interest also multiplies, and we screen, identify, and we get those lines where these the clones are, the recombinant clones, as we say. We basically select for that, first for the transformants and then for the recombinants. But when we are micro injecting, we do increase the number by cloning, but before injecting it into the mice, it is obtained in a linear form, and generally, all the prokaryotic vector DNA sequences are removed. So we prepare the DNA in the large number, we digest it suitably so that we can obtain only the gene of interest. We do not need the vector DNA sequences. So I hope you understand that why recombinant DNA technology was important, because when we want to clone a gene of interest, this gene of interest we are identifying and taking out from some cells, or eukaryotic cells or prokaryotic cells or organisms, we are getting it from there. But we do not get it in large number, we get it in small amount. So to increase the amount, we clone it into prokaryotic vector sequences and introduced it into suitable host such as a bacteria, so that the biology of the bacteria, which is the cell division, will be harnessed to increase the number of, or the copies of, this micro injected DNA. Now if you remember, in the advanced techniques we also talk about the polymerase chain reaction. So this polymerase chain reaction also increases the copy number of the particular gene of interest. So we can do anything, we can PCR amplify it, and then, but the main thing is that we need to get the DNA, the gene of interest, which is free from any other sequence or free from any other buffers or all the material that we have used it for in vitro cloning or in vitro amplification. So this micro injected transgene construct should be generally in the linear form and devoid of any other DNA sequences or any other material. So this DNA that we obtained, we then micro injected. So you can see this is a micro manipulator. This is a micro manipulator. This is a a system where we have a microscope, and in this microscope we can basically this is the platform where we keep our eggs. So we keep our fertilized eggs on the platform here, and you can see there are certain needles, and there are there is a micro injector, and then there is a a suction pipette which holds it on the place, and this entire system where we can visualize it. It can be visualized on the screen as well, that how this micro injection is basically done. So this micro manipulator setup needs to be there. Obviously this facility is not present in every lab, but now creating a transgenic mice is not that difficult, and many institutes have these facilities that outsource it for the other labs to produce it as well. So there are certain institutes which will have these facilities, and what a normal general lab would do is that they will isolate the gene of interest and probably clone it into a prokaryotic cell in vector, and introduce it into a cell to create the number of copies, and then give that DNA to the facility so that they can produce a transgenic mice. So this transgenic mice micromanipulators are not present in every institute or every lab, but they are outsourced. You can pay and get the transgenic mice made. So these are the micromanipulators, and as you can see, that is how the working can be done. So this is basically, again, in that circle, this is the platform where it is kept, and one can visualize it through the microscope and do the micromanipulation, or there can be certain screens as well where you can see large image of this particular area, and then you can micromanipulate it to introduce the DNA into the fertilized egg. So this is how basically the fertilization in the micro injection takes place. So this is a fertilized egg, and that is basically how the micro injection is occurring. So this is a plate, and you can see that it has been put into certain media. So these are the fertilized egg which has been put into certain media, and then they are covered with the 2 drop of oil are introduced from the either side, and then they form a film over this entire media containing the eggs on the slide. So this oil basically helps in the prevention of evaporation of the media, and the creation of, or basically the dehydration of the zygotes, so that and even the loss of zona pellucida does not occur. So this kind of oil film is also introduced so as to avoid the prevention or the loss of zona pellucida, or the degradation, or the basically the dehydration of the zygote. So the zygote remains intact in this media, and then as you can see, it is held in place by a suction pipette, and then micro injection is done from the other end. So this is basically a micromanipulator, and this is how the fertilized eggs are there, and then the introduction of the occurs. So we'll talk in detail about this in our subsequent sessions. Thank you.